Thanks very much again, Jim.
So, hi. Hi everybody. I'm really happy to be here. Thanks for the invitation. My name is María del Pilar Benites. I'm currently working at Instituto de Biología at UNAM, and I'm here to present the work we've been doing for last 3 years, entitled The Effect of Land Use in insect biodiversity and in tropical deciduous forest in Mexico. Hopefully English, my English will be clear. Okay. I don't know why it's not. Okay, here. Okay, so to start, let's talk a little
Sorry,
bit.
Pilar.
Yes.
Maybe you can try to share again the presentation because we are seeing the PowerPoint interface.
You're not seeing the presentation? Okay. Okay, I will try again. Okay, let's see. Let's try again. Do you see my my PowerPoint now?
Yeah, I think we are seeing only the first slide, not like the full view. Yeah.
Okay. So. Okay, let's see if this works.
Yeah.
It worked?
Yes.
Okay, perfect. So I was saying, let's talk a little bit about insects. They are one of the most common animals in our planet, and they represent more than half of all known species, with 1,000 species, around 1,000 species described, and it is thought that there might be almost 8 million species yet to be described. So they represent an essential component of the ecosystem equilibrium due to the different ecological roles that they provide in virtually every environment. Such ecological roles can include pollination, organic matter decomposition, predation, and prey interaction, to name a few. So over the last decade, insect decline has been increasingly been documented, and even when there's some debate about the extent of this decline in specific groups, they all agree that insects are declining in species, abundance, and biomass. So as causes of this decline, they have been pointing out climate change or ecosystem overexploitation and habitat loss, and they probably interact between each other in really complex ways. In insect decline, habitat loss has been pointed out as one of the main causes of its decline. So metabolic coding, as we've seen through this whole meeting, has shown the utility, how useful it is in biodiversity studies. Wait a minute, sorry, I need to move this. Okay, so in biodiversity studies, not only to build biodiversity inventories and the monitoring biodiversity in time, but also in ecology studies. Okay. Okay, so let's talk a little bit about the tropical deciduous forest, also known as dry forest. It represents one of the most diverse biomes in the world. This forest is defined by having a strong seasonality that causes an alternative deciduousness, with most of their trees losing their foliage during the dry season, followed by regreening or evergreen physiognomy in the rainy season. In North America, tropical deciduous forests, or TDF to shorten it, are mainly distributed along the Pacific coast, with some isolated patches in Veracruz and Yucatan. Well, like I said, the diversity in this forest is very high, and also their endemism is very high. And this forest has suffered a substantial contraction in its range, mainly due to human land transformation. And to date, few studies have documented how the anthropogenic disturbance has impacted the biodiversity and community assemblage of this type of forest in Mexico. So we were interested in studying insect species diversity at a community level in 2 regions of Mexican tropical deciduous forest. To that end, we started this project in 2021 in 2 regions of tropical deciduous forest, in the Estación de Biología en la UNAM y en el Parque Nacional Huatulco in Oaxaca. Our aim was to set a ground line for biodiversity evaluation and monitoring of insect communities in these 2 regions, and assess insect biodiversity and community composition, and how this biodiversity changes with respect of region, land use categories, and across month. And we also wanted to evaluate how environmental influences species turnover. So we started this project in 2021, like I said, as part of Proyecto Ciencia de Frontera grant. We had a really big team. This project is a collaboration between different institutes and organisms inside UNAM. And the leader of the project is Alejandro Saldívar Riverón, but we have different researchers and students involved to make this possible. We started this project during the pandemics, which was the biggest challenge because UNAM facilities were closed during that time. So we had to work around that, and it kind of set us back in time with our results. So we sampled these areas through malaise traps. We had in the two regions, in Chamela, we had 10 malaise traps inside the Reserva de la Biosfera, and 10 malaise traps outside the Reserva. In plots, in 1 hectare plots, that used to be agricultural sites and are now fenced and has been without human activities for a certain amount of years, from 15 to 30 years without human activities. So because there was a natural breakage in this, in the years they have been without human activities, we separate these sites outside the Reserva as medium ecological succession to talk about plots that had been 30 years since last perturb and low ecological succession for sites with 15 to 20 years since last perturb. In Huatulco, we didn't have this kind of plots and nor information about plots outside the Parque Nacional Huatulco. So we set 5 traps inside the park and 9 traps outside the parks in places close to the parks, but with no information, and places where there is actually agricultural activities going on. There are tropical deciduous forest patches around these agricultural fields. So sampling took place in Chamela from May to October 2021, while in Huatulco took place between June and December 2021. We kept going with our sampling, and we now have more results, but now I'm just presented results from this time range. Because it's what we already had processed at the time. So we also, in each plot, took ecological variables. In an area, in a circular area of 10 meters of diameter, with the Malay trap in the center. Within that area, we measure every tree with perimeter at breast height higher than 20 centimeters. We record which species that trees belong to, and also microhabitats in each tree, like lichens or invertebrates nest, or lianas or creepers, vines that could be on that tree. So we also measure vegetal coverage and the amount of deadwood matter that we find within that area. And for each region across month, we also measure climatic variables like temperature and precipitation. So each Malay trap, we replace the bottle every 15 years, 15 days. And then in the lab, we merge two 15 days samples in a month sample. So we end up with 218 samples, which in the lab were dried and side sorted, that to prevent insects with really big biomass to bias the representation in the sample. So everything larger than an Apis mellifera, we took like a leg and put it back in their sample, and the rest of the specimen was kept ideally to be incorporated in the collection. That was not always the case. So once we had our samples dried, we grinded in this grinder into fine powder, and we sampled 25 milligrams of that powder to be sent to extract DNA and PCR amplification. Well, lab processing, this image comes from Liu et al.'s paper of 2019, but it represents roughly what we did. So we had our bulk sample, we extract DNA, we amplified by PCR, CO1 in our case, since we're working with animals. We targeted 418 base pair sequence. We ligate adapters and barcodes. We multiplex and send these samples to sequence at Illumina NovaSeq. So once we got our results back, NovaSeq produces a huge amount of data. We started with quality filter, primer and tag and adapter removal of our reads. We demultiplex them into samples and we overlap our R1 and R2. And we end up with around 3,000 reads per sample, every read of 418 base pairs, which we process to try to detect contamination and also chimera removal. And we end up clustering by similarity at a 97% threshold. We assigned this motus, standing for molecular operational taxonomic units. We assign them to taxonomic identification using BOLD and NCBI databases. So I'm going to present the results we have so far. Altogether, Huatulco and Chamela's regions, we end up with 7,000 motus, which only 18% of them are shared between Chamela and Huatulco. The species accumulations curves shows that the slope is less steep at this point, and that probably we will need more than 400 samples to get to a good saturation of the place. By the estimations of Chao and Jackknife, we know we are around 70% of all species are sampled by our work. And we have like the most represented orders are Diptera, Lepidoptera, and Coleoptera altogether represent more than 70% of the motus. All our motus we could assign to order 99.7% of them, to family 93.74% of them, to genus 27.11%, and to species 13.86. So the amount of species we were able to successfully identify, it's quite low. So to look in detail, Chamela and Huatulco, we see the same shape in the accumulation curve and rarefication, and they are also the same orders were the most represented. And what we see is that Chamela has higher species richness than Huatulco. This is statistically significant through a GLM. And we also find that Chamela had higher richness in these medium ecological succession plots outside the reserve, while Huatulco showed no difference between conserved and perturbed plots in regards of its richness. So this is to represent what we found through the month. What we see is that there's a lot of change in species across month, and very few are shared between all months. It's not very easy to look at them in detail since there are 6 months sampling for Chamela and 7 for Huatulco. So to talk about now community composition, through a non-metric multidimensional scaling, we represent the plots in Chamela and Huatulco, and what we found is that there's a quite different structure between Chamela and Huatulco in regards of their community composition. And also, when we look at them as a correlation between sites, we see a structure between the two areas. And also, there's a considerably turnover because we couldn't find a correlation larger than 0.5 between any of the sites. So to look at more details in Chamela and these different ecological conditions, we see that perturbed sites, they tend to aggregate, conserved sites tend to aggregate and differentiate themselves from perturbed sites. And in perturbed sites, what we see is that medium or low ecological succession, they seem to be intermixed. So maybe that categories that we set are not natural categories. And we also look at how these community assemblages behave across month. And what we see is that they aggregate in month, and there's a considerable change across month, and that the month with higher rain and temperature, which are June, July, and August, tend to aggregate from the others. So same with Huatulco. And across month we see more gradual, not so separated in space as in Chamela, but also following temperature and precipitation. So to talk a little bit about species turnover, we use zeta diversity, which measures the amount of species share, but 2, 3, 4, and so on. Plots, considering all plots like a multidimensional measure of diversity. So what we see when we plot the zeta decline is that the amount of species share rapidly declines from orders 2 to orders 5, and almost very few species are shared from 6 plots on. But when we make a graph of zeta ratio, which is the probability of a common species to be retained, we see for each order that those few species that are retained are really common and are present in almost every plot. And in all plots in Chamela. In Huatulco, this is also true for all orders, but Hymenoptera, which shows no species shared for all plots, and points out to a complete turnover of this order across plots. So here we have the fit of this zeta decline to either exponential regression or power law regression. So this fit tells me if the turnover is driven more by a niche differentiation or by stochastic processes. If it is better to an exponential regression, turnover is driven by stochastic processes, while if it's the fit is better to a power law regression, turnover is driven by niche differentiation, which was our case. So to look at species turnover within these ecological conditions, we separate chamela zeta ratio, this probability of retention, we separate it in these ecological conditions: conserved, medium ecological succession, and low ecological succession. And we see overall the same pattern as we saw when we plot them all together. While in Huatulco, what we see is that in perturbed sites, we see that complete turnover in Hymenoptera, suggesting that perturbed sites are the ones where the turnover in Hymenoptera is greater. So when we plot them all together, chamela and Huatulco, we find what we expected, since there's so much structure in their communities, that for several orders we see a complete turnover between both regions. But what could be interesting is that Coleoptera and Diptera do share species across these two regions. So we tried to look at the prevalence of different orders, how they are affected by these different ecological conditions in land use that we were studying. This is still a work in progress. So we use this predictive GLM model for presence and absence of all sites simultaneously, the four-corner model. Which what measures is how each condition affects prevalence of each order. And we see all kinds of results. Some of them are pretty weak and different. We don't see a pattern across chamela and Huatulco, which was expected also because perturbed sites are not the same in Huatulco than in chamela. And what I would like to point out is something interesting is that perturbed sites in Huatulco affected negatively almost every order but Coleoptera. So we also look at how ecological and geographical variables influence species turnover. We did that through multi-site generalized dissimilitary modeling, which measures how turnover changes with these variables. What we should look at is the higher we model the variables through I-splines, and what we should look at is that the higher the variable, the slope of the variable, the higher the influence in species turnover. No, so we measure this for different zeta orders, trying to represent from a local range, which will be a zeta order of 2, to a regional scale. So what we try to do is try to go from a local to and broaden the range towards regional, and seeing how the variables that affect turnover maintain or change. So we see that distance between plots, which is a geographical variable, and tree diversity are important drivers at a local scale of order 2, while total deadwood for chamela and canopy coverage, both for chamela and Huatulco, are important drivers at regional scale. at order 18 and 12 respectively. And we also see like tree diversity in Huatulco influence on turnover started to be very strong at a local scale and weakens toward a regional scale. So we also find that ecological variables explain a large portion of the turnover variation in Huatulco, while in Chemela, a lot of the variation in species turnover remain unexplained. So some conclusions we have so far, what we find is marked differences in tropical deciduous forest insect communities between Chemela and Huatulco, even though they're the same biome. We are seeing that insect communities are profoundly different. Change in land use influences insect communities in both tropical deciduous forest regions, and that species turnover seems to be driven by niche differentiation. We also find that sites with either past or actual perturbation hold high insect diversity. That's something that we were surprised to find, that the richness was really high, even in really perturbed sites. So that's an interesting result that tells us that even a small strip of untouched, tropical deciduous forest can hold high diversity and can be very beneficial if we think about not only for biodiversity conservation matters, but also because high diversity is very beneficial to agricultural practices. Oh, sorry. Sorry. Okay, to end up with, we also find that different ecological and geographical variables influence turnover at different scales. And ecological variables seem to have a stronger influence in Huatulco species turnover. And also, the low amount of species that we could identify tells us that barcode data references is far from complete in this biome, and that efforts needs to be stronger in this data reference building. Well, and that's it so far. Thank you for listening. Sorry for my broken English. Okay.
Thank you very much. I think your English is fine, better than mine. So I think we have, Eduardo, we have a question to Pilar, Pilar from the virtual participant. Can you maybe read it or maybe pass the micro to the person?
Just a minute.
Eduardo, can you read the picture?
Sí, sorry, it's in Spanish, so I can translate it if you want. So they are asking about SETA diversity indexes, which is the appropriate size of the sample to be used. I tried to run it on 9 plots, but results I got were not conclusive. I assume that an n equal to 9 is very small to run this type of analysis. I run it with smaller samples than that, and yes, they're not conclusive. Like you want to have a robust n to get good results, and yes, the bigger the better, but you can also run it with small samples. What you are looking at the tendency of the curve, and in general, you can get some conclusions by looking at that. Like I said, like larger sample is better. And if you have problems, feel free to contact me, and we can look at them and see if I can help you to get better results.
Okay, thank you. Other question from the virtual participant, Edward? No? Okay. I have a question for Jeremy, just the discussion in general now about, any kind of presentation about the, trying to assemble those reference library. I want to assemble a library either with museum or with fresh specimen. Where can I find the funding to do that? Because maybe in taxonomy, okay, you can probably, answer a specific question, if you sort of, going to assemble the library for, from a taxonomy viewpoint. But if I want to assemble it from, from fresh specimen, unless it's actually embedded into your grant, I mean, it looks to me like this is a bit difficult, to get it funded, and we know that this is, quite substantial amount of money. So I don't know if you have any advice or any comment about this.
Good question. Well, I don't even know where to start really, except to say, so at the Center for Biodiversity Genomics, when I was there, they certainly tried to tap into as many different funding avenues as possible. And sometimes your application needed to be tailored towards that particular sponsor or foundation or agency. And there were some, I'm thinking of some agencies like that were very heavy in supporting genomics research, that kind of standard barcode library work, it just might not compete with the other projects that were being submitted. So in that case, library building, if it was a component, was just not a major part of it, or not a primary selling feature of what was being proposed. But then I think of some of the other competitions, we keep referring to the Smithsonian and the Natural History Museum there. There's these small pockets of money through the Smithsonian Institution Barcode Network that really favor reference library building. So, and then there's just a lot in between those two extremes, I guess. I would just, I think you always have to have something exciting and innovative and applied nowadays to, to make it to the top of a pile of applications. But I don't think that precludes you from also having a library building component in it. I don't know, like, what do you think?
I've been, because I will not have a job soon, I've been looking at grants also, and some of the NSF collection grants are probably okay because there's a huge component of collection. I mean, I know you already have a great collection, but if we were to find a way to justify more collecting, I hope, and then have the component that, on top of the collection, we will be able to make a barcode library. So I think Voita was talking about this also for ERC grants, like collecting for the sake of collecting, although I think we all agree is the best to do. you need to find a way to justify it as a recurring grant. And I have a feeling that barcoding, we rode the wave for a moment, and now it's kind of like coming down a little bit. So we could find ways to reactivate the efforts of barcoding in general, but it has to be an extra thing for the grant itself. Or at least this is what I have discussed with some people in places where there is no barcoding at all. And yeah, so I do think there is a way to integrate it into bigger grants, but it's not the express objective, let's say.
Maybe Jose, for the arthropods of medical importance, that's easier to maybe find some reference summary.
To get funding, you mean?
Yeah, for the reference.
I haven't come across any agency that provides funding specifically for this kind of task. I think it will be hard to get funded. I mean, my experience at Cenacid, which is the agency that had given me most of the funding that I have, and I'm surprised that you got funded for the metabarcoding, because I know they are more into support more applied research than basic science questions. And so that was really good that you got funded through Cenacid. But I haven't really seen anything in NSF, NIH, specifically to fund building collections and references libraries. So.
Okay, thank you. So, Scott, I think maybe Eduardo, if you could habilitate Scott.
Oh, hi. Sorry, my camera is not working, but this is Scott Miller. There is no magic bullet to the funding question, but I also think that most funding agencies theoretically respond to the communities that they serve. And it does seem like a group like this that you have assembled has the opportunity to try to make the argument back to Cenacet that Panama needs reference libraries as well as capacity to use these kinds of tools for, the whole range of projects that we've heard about today. Other funding agencies as well, but certainly the group assembled here can make a lot of good arguments for why Panama specifically could benefit from having strategically targeted reference libraries in groups that are important. So that's all.
Yeah, okay, thanks. Milan, you want to comment about this, like from the viewpoint of Cenacid, like you think you might get something from them if you ask them for reference library?
Yeah, just my experience is fairly limited, and of course there are we could do better with more coordinated international efforts, and there are some projects which are trying to do that. But for example, this project which Alejandro Zaldivar is leading, which Pilar was referring about, was actually one of the few barcoding and meta-barcoding initiatives in Mexico over the past year. One of the few projects which got funded in basic science and in ecology as such. And they've been, from the proposals what we see, there are always some focused attempts on barcoding some economically important groups or some endangered groups, but they get rarely funded. And because it's being seen not probably edge-cutting science anymore, or sometimes maybe too basic science. And on the other hand, I have to say that these isolated efforts often or sometimes it's obvious that they don't have like long-term planning in terms of how the specimens, how the types should be treated, how they should be stored, how they should be made accessible, not only regionally and nationally, but internationally. But again, maybe it's probably not the best parallel, but as we put in our projects the money for publications and for publishing in open access journals, in some of these initiatives, we should get used to really putting certain percentage of funds and time, effort into building these reference libraries and really create some better framework where to feed these specimens and where to store them. Yeah. The same the Smithsonian is trying to do, at least in US.
Well, okay, thanks. Eduardo, from the virtual participant, do we have any question? No? Here in the audience, don't be shy. You can also, have a question in Spanish if you want. I have one question for Jose. I mean, we saw this today that we can have interesting insights into the microbiomes of those insects like the ticks, but we had, an interesting presentation about the dung beetles. Could we think of other, insect group where we could quickly get an idea of the microbiomes and then maybe because that's, a mind-boggling also diversity. So I don't know if you have any thought about this.
You mean like using bacteria to identify insects, or just looking at the microbiome? Well, in the case of mosquitoes, for example, we know that when the mosquito is infected with a virus, the virus replicates in the midgut. And so when there's certain bacteria with the virus co-infecting the mosquito, the bacteria can actually take resources from the virus and inhibit the replication of the virus. So certain bacteria can inhibit the viral replication, and as such, the mosquito become a poor vector. And so that's why I'm actually interested in that kind of relationship. I think bacteria are really important for other type of insects because they help the insects to digest certain type of food. And so I think that could be another way to look at it, like whether the bacteria that is associated with the insects helps out in, certain type of habitats to digest certain substrates. Or, I mean, there's a bunch of other applications that you could use the microbiome for. I think the protocols are pretty straightforward, and at NAOS with Kristen and folks at NAOS, we were able to do this pretty easy, I guess. And we have also applied these tools to look at specific species of mosquitoes. So I think that it's pretty straightforward already to be applied to other insects. We just have to define what kind of questions you want to get at, but yeah, I see that the sampling that you do with pitfalls, Malaise traps, and these other techniques could, provide samples to compare, looking at the microbiome. And another question that I have, for example, is whether the microbiome of an insect changes across seasonal. Like since Panama have this strong seasonal component, the dry season and the rainy season, whether or not that can change because of the season. But yeah, I guess there's plenty of opportunities to apply metabarcoding of both bacteria and fungi to look at species interactions and things like that. Was that the questions you had, or maybe I missed it out?
No, I think in part, but I was thinking like if you're interested to really get an idea of the diversity of all those microbiomes, instead of sort of randomly sampling bacteria everywhere, you can use the insect to really, do we have certain groups where you think that, might be better to sample because they might concentrate a lot of bacteria? This was sort of the sense of my question.
Well, I guess insects playing different ecological roles will be a proxy of different bacteria communities. And so, insects are found in water and soil. And so yeah, that could be a proxy. So insects could be a proxy of the bacteria that are associated with that particular group of insects. And yeah, indirectly, I think, I guess it could be used to assess diversity of microbes. Yeah. I mean, Kristen can maybe have some ideas about this.
I have seen some studies about looking at microbiome of ticks and sandflies to kind of like track the spread of leishmaniosis and the Lyme disease. And so I think for diseases, I think it's actually proving quite useful. And there's at least one study that actually sampled dogs in Brazil to try and trace leishmaniosis to then kennels in the US that are like, pedigree breeds. And as far as I understand, leishmaniosis goes through the mother to the kids, and it happens for all these kennels that have endemic or dogs from endemic origins. And I think it's just through metabarcoding, like they just grab either the dog blood or the sandflies, and they can find all these diseases, which I think funding-wise, I think human disease can be a good thing.
Yeah, but I'm not interested in this. I mean, I leave it to Jose and another person who can do that much better than myself. Eduardo, we have any question on here from the audience? Jose?
Just wanted to take advantage of the opportunity of having such an amazing audience here. And I wanted to pose a quick question about what is your take on training the next cohort of taxonomists? Because, there's this debate about whether we should focus on molecular techniques solely, or we should train people so that they can embrace both morphological identification and molecular stuff. This is kind of a challenge that we're facing moving forward in entomology, and I just wanted to know what are your takes on this?
Milan actually explained, in your talk, you were playing for actually having, a good collaboration between taxonomy on metabarcoding. So I guess, of course, this is excellent to see that at the Maestria of Entomology in Panama, you're trying to do both, which I think it's clearly the way to go. But maybe many people might not be of the same opinion. And of course, taxonomy is still regarded as very obsolete, science, and then it's difficult. It's also part of it is that little by little, I think, in terms of vertebrate and even probably plant species, we basically know what are the most common species, all the species on the planet. As far as we're concerned with insects, like we've seen that in many of the presentation here, we still have, very large share of biology to investigate. I must say, insect study or arthropod research in general is vastly underfunded. So it's something which is totally neglected. And I think, in terms of taxonomy, what kind of new description you see now? It's all basically insect and maybe, fungi, of course, but that's, or the microbiome, but that's also, using totally different techniques. So the sort of entomology can be also regarded as an obsolete science because you have all this taxonomic component, which is, incredibly important. If you want to know your bugs, you need to place a name on them and know to know what they're doing. So yeah, it looks like you should have both approach, and then that's certainly the way to go, and then that's excellent that the Maestria is really investing effort in trying to do that. What's the proportion, for example, for you? The question is, what's the proportion of the students who come to the Maestria which are still interested, maybe in taxonomy per se, as opposed to maybe more applied, aspect of entomology and maybe the molecular aspect? Can you give us some figure?
Well, I guess we give students sort of like a general formation in entomology, and we try to embrace, not only the old school techniques, but also the new tendencies. It's hard because you need to have the proper infrastructure and the supplies to, for example, train people on molecular stuff. But, since you guys were worrying about the type specimens, and, I think is really important to try at least to train an entomologist so that he knows what are the opportunities out there. And depending on the question that he wants to tackle, he could go, at least have the opportunity to branch out to molecular, or to stay in traditional morphology, in collections and stuff like that, or combine both and do other kinds of studies. So it's going to be hard to train this new cohort of scientists working on entomology, but I guess we just need to try to guide the student so that he have the tools, whatever the question he wants to tackle. And then that's the challenge that I was thinking about.
Okay, thank you. Any other question in the audience? No? From the virtual participant? No? Yeah, I think that's pretty important. I mean, you don't want to sequence your own sandwich in the lab, not knowing this. This is the problem. That might be a bit, easy to say, but when it comes to entomology, and then we have this incredible diversity, that's pretty important, just to be able to place name. And then we've seen the tendency in all those recent article about metabarcoding, addressing insect communities. We see the result only at the level of orders, and which are not terribly interesting, and then we very rarely you can go to the species level and really see what they're doing, because of the challenge basically with having the correct reference library, so that you can get a bit deeper into this. But next tomorrow, I think we'll have a set of, again, 8 different presentation going to be fairly interesting. I don't think we'll talk too much about taxonomy, but more just about the different techniques that we can apply to the soup, and then just there's a fair new bunch of methods to, for example, delimitate those bean or motus or otius or whatever you call them. And I think that's going to be very interesting. I think unless there is really last set of questions, I think we can close the meeting for today. And I would like to thank everybody who attended to the meeting, here presentially or virtually. And so we'll start again tomorrow at 9, and this will be basically the same setup on the same timetable until 4. Thank you very much.